nobody has a problem with that. In case we can share it with some people. In case you don't know who I am, I'm Diana Haberkamp. I'm with NF West. I'm the director here. I don't have anybody in my family affected with schwannomatosis, but I do have a husband and two children with NF type 2. So I want to thank you for joining us. We did put this out as coming from the NF Collective. So if any of you want to know what the NF Collective is, we can ask me some questions about it later, but it is a group of organizations working together. And I thought schwannomatosis was a great way to go and let's all work on this together since you are a smaller community. And then Vanessa was going to present this at our info fair, but we didn't have any schwannomatosis people there. So I said, let's get it online and get it out to everybody. So I'm going to introduce Vanessa, but I think most of you know who she is. So Vanessa is a health services researcher at Mass General Hospital and assistant professor of neurology at Harvard Medical School. She serves as director of research at the Mass General Family Center for Neurofibromatosis and Schwannomatosis, where she conducts research to improve the accessibility, quality, and patient-centeredness of clinical trials and clinical care for all types of NF. You can tell I was reading that, right?
I know, I gave you a long thing.
You did give me a long thing. So we'll keep it kind of casual, and I'm going to go ahead and let Vanessa take it away.
Yeah, I know it's a small, intimate group, so please feel free to interrupt me as we go with any questions, either for you or things you think I should explain better. When people watch this on the recording, I really value your feedback. So as Diana said, I'm based in Boston at Massachusetts General Hospital at our NF clinic here. And I've actually been working at the NF clinic since 2010. So I'm coming up on my 15-year anniversary. And for most of that time, I have been researching really all types of NF, but including schwannomatosis. I actually did my PhD dissertation studying why it was taking us so long to diagnose people with schwannomatosis, what was going wrong, and some potential ways we could fix it. So this topic is really near and dear to my heart, trying to find treatment, having been able to talk to people for so long about how this really impacts them. So I'm really happy to share with you today kind of the story of how our knowledge about schwannomatosis has grown over the years and every way that we've been tweaking our clinical trials and planning and learning from them to really try to get to the point where we have a drug that works or multiple drugs, ideally, that work to really help people with their pain. Oh, I see I forgot to change my title here, but we're giving this talk. This is all accurate as of February 25th, 2025. So I only have one slide of background because I bet you guys know this even better than I do in terms of why is this important to people with schwannomatosis and why are we so focused on pain treatment. And so this is actually some of the earliest work I did with this community back in 2012. We published this. This was really the first large retrospective study where we looked at 87 schwannomatosis patients who had come to Mass General Hospital over the prior decade or two to really even start to get a sense of like, what does this disorder look like? What's happening for folks? And what we found was about 2/3 of people with schwannomatosis reported that they had chronic pain, and it was often pretty severe. So about 18% of people in our cohort had said they were even unable to continue working because of this pain. And 80% of people, so the vast majority, had had surgeries in the past to remove painful schwannomas. But unfortunately, we saw that surgery wasn't always effective. It might be effective in the short term, but not the long term, or it might not actually be effective at all. So really only about 1/3 of the people, at least in our sample, who had had surgery said, after surgery, I'm completely pain-free in that area now. So we knew that there was still a role for medications to help manage this pain where surgery couldn't be used. And we did see a lot of folks were using it. So again, about 2/3 of people were trying a different majority of medications. Most people using maybe multiple medications, but still having these problems. So I think it was really clear at this point, right, that this is a high unmet need. This is something that needs trials, but we had a lot of limitations at that point. And how can we design these trials? we didn't really have a good numerical kind of precise understanding about what the natural history is, who has pain, how does it change over time, what's the impact, which makes it hard to plan clinical trials where you need kind of those specific numbers to really estimate how long you need to study things and how many people you need to enroll. So kind of the first part, or I guess that was the first part. This is now the second part of our story was, okay, let's understand what does pain look like. Let's put the numbers on it to help us plan trials. So moving from that 2012 study, now we're at 2015. We started a natural history study. And so we recruited people from the International Schwannomatosis Registry. I think this might have been prior to or pretty early to the NF registry. So there used to be, and I'm not sure if it's still running, a specific schwannomatosis registry to help find patients across different clinics. And we recruited 79 adults with schwannomatosis from 4 places. So here at Mass General in Boston, at New York University in NYC, Johns Hopkins, which is in Baltimore, and then the University of Manchester, which is in the United Kingdom in England. And so this was a natural history study. That means we didn't give anyone treatments. We just said, can we measure your pain over the next 5 years? And so people completed online questionnaires every 6 months for up to 5 years. And the primary thing that we were looking at was how bad is your pain? And so I'm going to show this because this comes up a lot in clinical trials. This is often the primary outcome or kind of the main metric of success for trials. And it's really what you hear at the doctors every day. How bad is your pain on a scale of 0 to 10? And we specifically ask, what was your schwannomatosis related pain, and what was it at its worst? Thinking that, people can have other kinds of pains, headaches, I don't know, you take a fall and sprain your ankle. Excluding that, thinking about what you think is kind of related to schwannomatosis, how bad has it been? And what was the worst? What was the absolute worst you experienced this week? And so I had mentioned we had enrolled 79 people who were filling out our online surveys. About a little over half were female. The median age show in the middle was about 51, but a wide range, so people who were 30 to 78. And we found that more than 50% of patients had pain that was a 5 out of 10 or more. So on that 0 to 10 scale, 5 or more, which we often call kind of moderate to severe pain. So pretty significant pain. And this is at baseline. So when they first signed up for the study, 77% were using pain medications, but it didn't really seem like any of the existing pain medications were particularly more effective than others. So you can see here in this graph, I'm going to make my little laser pointer. This is kind of an overlapping Venn diagram of all the medications people were on and multiple medications. So for example, in this part here is people were taking NSAIDs and neuropathic pain medicines, or NSAIDs, neuropathic, and opioids, so 3 or more medications. And you can kind of see from the numbers, it's not like one class stood out. It's not like everyone is saying, oh, we're all on NSAIDs because like that's the amazing drug. Everyone was kind of trying everything. Different things might be working partially for different people, but nothing really jumped out as this is what people should use. So kind of disappointing. It would have been great if we could have found out what was effective, but really solidifying, we're going to need to find new stuff and we're going to need to design good clinical trials to do so. And so we learned 2 really important things also from this study that would help us design clinical trials. One was what should we compare against? How good are existing medications and so that we know what's kind of the bar we have to set. And so I'll walk you through this graph. This is a graph of kind of the different months on the study. So 6 months, a year and a half. And this is showing how many people got better, said that their pain got better. And we define better as 2 points or more. So say going from 10 to 8 or less, or going from 8 to 6 or less, because that's like something we're confident in. We think that's a real change. And you can see here how many people got better. So when we asked at 6 months and said, compared to when you started, what's your pain? About 30% of people said, my pain's better. And this slowly ticked up over time. You can see at a year, we're about 35%, and then it kind of levels off. You can see just above 50%. So about half of people said that they had at least some improvement in their pain, but half of people didn't have any. It was the same or worse than it was when they started. And so that kind of helped us get some information. And the other thing it helped us see is, we really have to be thinking about how durable these pain medications are. Because yes, maybe 50% of people at some point felt better, but when we tried to look at how well was that sustained over time, we saw the performance was a lot poorer. So I'll walk you through this here. The response at 6 months, so it was actually 29.5. And at 1 year, we were up to 36%. So 36% of people said that it was better, but the number of people who said they were better at both time points, so at both 6 and still at 12, was this low number, 16%. So I don't want to overwhelm you with like the graphs and the numbers. Maybe that was a little too much. But I mean, the message being, it's very clear that we need to find things that are going to be durable, that are going to last. And it has to be kind of greater than this number. So if we do measure something at 6 months, this helps tell us, well, if it's at 30% of people respond, that's not that much better than current. But if we can get 60%, right, that would be a real benefit and different than what's currently available on the market. The other thing we learned was kind of what should we measure? How is pain affecting people? And kind of what are those relationships? And I'll say this is also a complicated figure, so I'm going to walk you guys through it. And I would love actually your feedback of what's missing here. But that number that I was just talking about is what we call pain intensity. How intense is this? How severe is this on a 0 to 10 scale? And we wanted to map out how does that pain then affect other parts of people's lives? And that gives us the sense of what else do we need to measure in a clinical trial to show if a drug is having a benefit. And so here on the left are things that we think influence the pain, how severe the pain is. One of those things is your genotype. So there has been published literature that suggests people with LZTR1 mutations tend to have worse pain than people with SMARCB1 mutations. Of course, there are people with LZTR1 that don't have pain, and SMARCB1 patients can have pain. It's not a total yes, no, but it tends to be higher in that group. So we think there's something happening with the types of mutations people have that might be influencing how much pain they have. Also, pain quality. That's the word we use for how does the pain feel? Is it stinging, burning, aching? Is it more neuropathic, that kind of nerve pain? Or is it more what we call nociceptive, kind of that achy pain? The different ways that people feel their pain can influence how they, perceive how bad it is and how intense it is. And then, of course, what we've just been talking about, use of pain meds. If you're using an effective pain med, your pain isn't as intense. If you're not using an effective one or none, maybe it's worse. So that is things we have to consider. And then we also look kind of downstream. What is pain intensity effect and what are the things we should be looking at? One of the major ones here is pain interference. You might see that phrase a lot. That's often a secondary outcome or co-primary outcome in trials. And that's how much does pain interfere in your day-to-day life? So some people might say my pain is a 6 out of 10, but they are still going to work, doing their activities. And another person, they could say their pain is 3 out of 10, but it's really, changing their lifestyle, their work, their school. And so we want to measure both of those aspects, the direct number, but also what's the impact on your life. And then as you can see, there's a lot of things which I'm sure is no shock to the people on this call that pain affects. It's been shown to have effects on your mental health, your sleep, your physical functioning, and all of these things are interrelated, which is why this diagram looks so complicated and the arrows keep going in all directions. They're all interrelated, and so kind of mapping out these interrelationships helps us understand what to change. Any questions on this before I go into our first trial? And I will say this is a conceptual model. This is kind of what we're currently thinking. It's not set in stone. I'm sure there are other factors I'm missing that are also related, right? All of the different domains of our lives are interrelated in pretty complex ways. But I'd love to hear now or later, yeah, if you guys think there are other major things that we should be measuring that aren't on this list. I would love to know. Please feel free to share. So kind of to put us in time, that was now running from 2015 to I think it's at 2019. And we now had all of this great data. We had all these numbers and we could plan for a clinical trial. And so then we get to, I'm going to say 20, I think this is 2019. We opened the first clinical trial for schwannomatosis related pain in terms of a drug trial that I know of, and it was for tanezumab. And this study, I should say I think every study I'm about to talk to was led by Dr. Scott Plotkin here at Mass General Hospital. So he was the PI of this trial, and he kind of brought this to the fore in terms of medical treatment. And so thinking about where we were in the world in 2019, right? A lot of attention was being paid to opioids and the opioid epidemics. So we knew a lot of people were looking for drugs that were not opioids. So really driving discovery for pain treatments that didn't include opioids for both schwannomatosis and the larger community, of people with pain. And we also had found out from a variety of preclinical work that something called nerve growth factor seemed like it was relevant to schwannomatosis pain in specific. So we found elevated levels of this nerve growth factor in schwannomas that had been resected from people with schwannomatosis, so taken out in surgery, and then studied in the lab. So we thought this seems like a good target. And I'm using the royal we here, I should say. I don't do any work in a lab. We have wonderful collaborators like Lei Shu and people from all across the country who do this wonderful work in the lab. So we as a community found this out. And then the last piece of the puzzle, knowing that there was nerve growth factor might be relevant, is we found a drug that targeted nerve growth factor. That drug was called tanezumab. It's an antibody that specifically targets nerve growth factor. And it had actually been very widely studied and was FDA approved already for the treatment of other pain conditions. Well, actually, I'm going to take that back. I do not think it was FDA approved, but it had already been studied in clinical trials for much broader groups of people with arthritis. So there was a lot of safety data. So that's one of the reasons we often like to repurpose drugs that are already being studied, because then you get to benefit from a lot of the initial pilot work that drug companies and others have done to prove, what dose should human beings take, what's safe, and not going to have really bad side effects. So we were able to kind of capitalize on that research, bringing tanezumab forward into the schwannomatosis community. And being the first kind of drug trial, we had to think about how to design this trial in a way that would balance the need to have really rigorous scientific data, but make sure everybody ethically, we wanted everyone to try this out. And so I'll walk you through this because it happens in all of the trials I'm going to talk about. We use something that's called delayed start randomization. So this means that we have two groups of people in the trial, and for the first period of time, half of people get the active drug and half of people get a placebo, so an inactive pill. And it's a double blind, which means the patient doesn't know which of these two drugs they're taking, and we as the researchers don't know which drug they're taking. That way it doesn't bias either of our opinions, because there's some concern when you're a regulator, so someone at the FDA looking at this data, you might say, well, people knew they were on placebo, so they just assumed it wasn't working, or they knew they were on drug and were hoping it would work, so maybe they biased their numbers a little down. So this way, there's no doubt. You don't know, we don't know what you're on, so the numbers are kind of that more pure, unbiased number, and that's kind of the information that the FDA and others really want to see to know if it's working. But in addition to that, we said, well, what if you're on the placebo group? You might, that's not really a good incentive to be on this. You don't get to see. So let's have this second period where everyone gets the drug of choice. And so this is how both the tanezumab trial is designed and the Starfish trial that's currently ongoing that I'll talk about. And so that's how we've kind of handled this. And I'll walk you through, this is kind of the study diagram we have. So this part here is what I just showed you, that we have a double-blind treatment where some people get tanezumab, some people get the placebo, and then a single-arm phase where everybody is getting tanezumab. And then we followed people for 6 months just to make sure there was no weird side effects that came up, right? Like that everything looks safe. And here's kind of, I'll walk you through, this is the timeline of kind of what happened. So first here is number of visits. You can see there was 12 visits total, although a number of those visits, the ones with stars, were telephone visits. So there was actually only, it looks like 1, 2, 3, 4, 5 visits in person over these 12 visits. And here's the days. So if this is day 1, when you take your very first dose, this is day 15, 29, so kind of 2 weeks, another 2 weeks, and then kind of every 4 weeks after that to follow folks up. And the way this was dosed is actually, I think it was only a single dose, and that lasts, like the effect would last this whole time. And then you could have the second dose here. And so our primary way of looking at this was what happened at this second dose at the end of the double-blind study. So that was the plan. And the folks that we included, so it was pretty wide. This was people who had a clinical diagnosis of schwannomatosis. You did not have to have genetic testing. You had to be an adult, so 18 years and old or older, and you had to have moderate to severe pain. Some key exclusion criteria, we weren't able to include people who had arthritis for safety reasons because there was some concern about whether this drug might make arthritis worse. And you also had to discontinue taking other NSAIDs because this is an NSAID and you can't take 2 of those same at the same time, which makes me realize I forgot to say an important point. This trial and also the Starfish trial, we didn't say that you had to come off of your other pain medications. So if you were on a pain medication and yet still had pain, you could enroll without having to come off, because kind of that would be then the endpoint would be adding this drug to your current regimen, make that effect. With the exception of NSAIDs, like I said, because we didn't want to give you too much of those medications. So people did have to come off of NSAIDs or not be on them at all to be able to enroll. And let's see, we were able to find 52 people. So this is about kind of screening subjects. You can see where they came from. We talked to, so 52 people with schwannomatosis or who were interested in the trial. A large number of them came from our clinic here at Mass General Hospital, but also from other institutions who referred us patients, from the Children's Tumor Foundation's NF registry where people had registered, people who found us on the internet through clinicaltrials.gov and Facebook, so different ways of connecting with folks. But unfortunately, we were only able to enroll 9 of these 52 people. We had been hoping to do, I'm wondering if I said it somewhere. I should have. I think we were hoping to do 46. Oh, here, I put it here. We were hoping to get 46 people. We only got 9, which was kind of a disappointment. The major reason for this was COVID-19 pandemic. So I talked about how we were planning this trial in 2019. We opened finally in October, I think I said it here, October 2020, because we had to delay for the beginning of the pandemic. But then even after we were allowed to open trials again later in 2020, you all remember what it was like. It was not a time people wanted to be flying across the country to participate in a clinical trial. We were still, that was a time where we didn't even have vaccines yet. So that was the major barrier, and it was a really unfortunate part of timing. But it wasn't lost data, which is the whole reason I, talk about this trial still. We really learned a lot about running this trial and what it was going to take for us to successfully recruit. because we also systematically looked at kind of what are these other barriers that are happening. And I'll categorize them here to know. There are some that are kind of inherent to schwannomatosis. It's rare, so there's not that many people to find. A lot of people might not have had a formal schwannomatosis diagnosis. So maybe we really thought they had schwannomatosis, but they had never had a biopsy or surgical removal. So they had no tissue to actually prove that they had schwannomas, which is required to get a diagnosis of schwannomatosis. So challenges kind of with people not having the diagnosis, so not qualifying. We also had kind of issues with our study design. So I'll skip to this one, time, right? The perpetual issue of time, travel, finances. Even though I said a lot of our visits were telephone visits, there were still, I think we just said 5 in-person visits. That's no joke if you don't live in the Massachusetts area to come in and do, right? That's a huge burden. And so obviously that's off-putting when there's a single center trial that's not close to you. And like I had mentioned about this particular trial, or actually about the design, you had to keep a stable dose of your other medication so that we could isolate the effect of the new medication. But it was only for kind of a short benefit, right? Well, I think I should have calculated the days into months, a few months. And so a lot of people told us, you know, I interested, but like, what if it works and then I can't get it anymore? Or like, I have to get off the NSAIDs and then my pain gets worse, right? There's a lot of trade-offs people are making when you're asking them to change their pain medication dose and how is that going to affect their life? And if it's only for a few months, is it worth that disruption to do that? And then, as I mentioned with the drug, this particular drug had some limitations around not letting people be on NSAIDs or having arthritis. And it turned out actually a lot of people we screened had arthritis and they couldn't participate for that. And the reason I go into all these barriers is because it gave us ideas of what to fix that we have now put into practice, is the like take home message. We can't fix this, that schwannomatosis is rare. That is what it is. But we can fix a lot of the other stuff, right? So there could be efforts, right, to help people get a diagnosis who need one, upstream care. We can design the trials to have less travel, and we can give financial stipends to help with financial burden. So this is a big effort with, you might have heard the term decentralized trials. For a lot of the folks in RAINS might have heard that, right? Doing things closer to people's homes or having them do some things at home rather than traveling to clinic to reduce this burden. And of course, new drugs, finding different drugs with even better safety profiles that might be more attractive. And I'll come back now to this last one I skipped, which is about what about people who don't have moderate to severe pain or who don't look like on the measure that they have that pain? We learned a lot from this study about how complex schwannomatosis pain really is. And so I'll show you, this is just 2 sample people, one person in blue, one in orange. And we learned a lot about how different some people could have fluctuations or steady kind of pain. So you can see this person in blue has many weeks where they have exactly the same pain week to week, fairly stable, some changes, but if you measured it on this day or this day, it didn't matter, you'd get the same number. There are other people who had very, high fluctuations in pain. You can see here up and down and up and down. And we learned from this that it can be really important how you measure pain. So I'll give one example. This is day 0. That's when we were screening people. And day 1, the first day of medication. This person had a 10 out of 10 pain on day 0, and the next day had 3 out of 10. So one day different in asking how their pain was, you would get a radically different picture if you only asked on that day, right? So if you had only asked on this day and they said, I have a 3 out of 10, you would have said, I'm sorry, you're not eligible for this trial. But if you had asked one day before, you would have said 10 out of 10, you definitely should enroll. And same thing in terms of measuring the effect, right? If we had started here and then went and measured out to here, you'd say, well, 3 and 3, nothing happened. But with one day difference, you could have said, oh, it was a 10 and now it's 3. This is a miracle drug. And so one of the lessons we learned is that you can ask people just on one day, right? A lot of people are going to have this fluctuating or intermittent pain that makes it look like they are not eligible or like that they don't have enough pain, but they actually do. And so we've specifically incorporated this into future trials where the screening is not a one-time measurement. It's now measurements over a longer time. So I'll mention this on starfish. It's over a whole month so that we can get a better picture of what does your pain look like, and then we can really tell if it's improved. So that might be actually something I bet to all of you is so obvious, like especially if you had pain that fluctuated like that, you might be like, well, duh, you could have just asked one of us and we could have told you that. But this is the learning process kind of all of us researchers go as we're trying to figure out how to measure. And like I mentioned, we're also now doing some of the kind of things to address the other barriers we experienced in the tanezumab trial. So let's incorporate more telehealth visits, things that people can do at home so there's not as much burden to participate. And are there ways that we can extend people's access to drug beyond the trial so that if they are having a benefit or think that they like it, they can continue getting it in some other fashion? That way, there's more incentive for folks to join. And so all of this really informed the current trial that is running that I want to talk to you about, which is called STARFISH. It's an acronym for Screening Trial for Pain Relief in Schwannomatosis. We took a little liberty with the letters there so that we could have an acronym that made sense. And I'll tell you a little bit about how this is designed. So this is also, I know, another complex figure. I'm going to have to do a better job breaking them down in the future, I think, because it looks complex, but hopefully we can simplify. The idea in a platform trial, which is what this is that we take a group of people who have schwannomatosis and pain, and we have multiple different drug arms, two that we have running now, but also more in the future. And so rather than standing up a single trial for each drug, and then the trial's over, and then you have to go through all the paperwork and stuff to set up a brand new trial to study the second drug, and then that's over. And then after that, you set up the third drug. Now, could we do them all at once? One set trial, which we call like the master trial or master study that people consent to. And then as soon as new drugs are ready to study, they can be kind of plug and play into the trial. And that way we can kind of speed up access to this. It's also really good in that there's common eligibility criteria and endpoints of what we're measuring across every trial so the results are much more comparable, right? If we're pulling from the same pool of people who've signed up who are testing each drug and we're measuring the success of that drug exactly the same way, it becomes easier to say, even though these are not head-to-head trials, which ones are working better. And that common infrastructure here that I said makes it more efficient, right? Rather than standing up a new trial, it's already running, and so it's a lot easier to put in. So for this currently Starfish trial, we have two drugs that we're studying simultaneously. There's the first drug arm is siltuximab, and it's an infusion. So that's why it kind of looks like this. And then the second drug we're studying is arenumab, which is a injection, kind of like if you have heard of the Ozempic or those GLP-1s, kind of a similar injection. And we're measuring the effect of these drugs. And then hopefully right now there's not a drug C, but the plan is if soon as we find something that would be a good fit for drug C, we can plug that in. So this is kind of a perpetually enrolling and expanding trial. And so we're really hoping that this trial design is the first step, something where we could rapidly and efficiently screen for whether drugs have pain relief. So really the motto for this trial is how can we figure out as quickly as possible the kind of go, no-go decision? What is worth taking to a bigger, more formal trial and what is, obviously not going to work. And so this is exactly the same as what I showed you before. It has the same design as tanezumab, At the beginning, half of people get randomized to take active drug and the other half get placebo, and neither us nor the patients know which one they're on. But then in the second 12 weeks, everybody gets active drug. So everyone at least gets to try it out and see what it is. And that way, everyone gets a chance to see what their experience would be. And they'll know that everyone will know at that point that they're on this medication. And so this is also a short trial, 12 weeks of active treatment, 12 of this open label phase, and then safety follow-up. And so a little bit about the design in case other people are interested. We're looking to enroll 40 people is what we're thinking who have pain, who want to be in this trial. And for each of these sub-studies, so each drug, we're looking to enroll 20 people. We think that's the signal we need to kind of quickly ascertain, is this something that looks like it's going to work or not, right? And to be on the study, you need a clinical diagnosis or molecular schwannomatosis, molecular being like genetic testing, but you don't need genetic testing. You could have also a clinical diagnosis of schwannomatosis, again, an adult having moderate to severe pain, and just a couple of other kind of just specific things. And then each drug might have its own indications based on what would be safe for folks. And so to talk a little bit more about these two drug sub-studies that we have been discussing, the arenumab and siltuximab, I want to go back a little bit to the biology, and I'll say this as the caveat. I'm not the person in the lab doing this. I'm really glad we have a great partner. So actually the co-PI of the Starfish trial is Lei Shu, who works here at Mass General and does a lot of this preclinical laboratory work to identify what's happening. And she had found that in these models, so these lab models where we have schwannomas in dishes or in mice, that there are a lot of number of cytokines that are overexpressed when there seems to be pain. And this is kind of a little model. So you can see actually NGF, nerve growth factor, which happens when there's inflammation or injury. That's what tanezumab was targeting. But further downstream, there's kind of these other molecules that were in that same pathway. And I'll point out two in particular. So siltuximab, which is the first drug, that infusion drug, is a drug that had already been approved for something called Castleman's disease, and it targets IL-6. So one of these other cytokines that's like NGF that we're thinking is involved in pain. And the way that we experience this pain signals as it goes through our nerve up to our spinal cord. The second drug is arenumab is the name, and Aimovig is the proprietary name. And this is one that works on CGRP. So another one here where the transcription is higher. So they're targeting two different agents, but all in the same pathway, same as with NGF that we think is important in schwannomatosis. So hopefully to work. And so the assessments that we're doing, we're following folks, as you saw, over that time period. The primary outcome of the trial is this change in pain intensity. We want to know, does this work? Does this cut down people's pain intensity? But we're also studying, we want to take the opportunity of having these folks, we value everybody's time coming in to really learn a lot about this. So we're also going to assess the safety. So how safe is the drug? How tolerable is it? So looking at, adverse events. So some clinical labs and physical exam just to make sure everybody's tolerating this well. We're going to assess quality of life. So we have a number of surveys planned, filling out, and like I had shown in that concept map, pain interference, physical functioning, depression, anxiety, social aspects, are really trying to get a holistic picture of what folks are doing. I actually don't think we're doing qualitative interviews anymore. But hopefully maybe could add that in to really learn and talk with you guys in depth about what it's like. Probably would have been helpful to talk to you, David, and find out what you just said and get you an answer on that earlier. We're also measuring what other pain medications people use. So could they come down and dose on their other medications while on this as part of the change? We're also asking folks if they're willing to donate blood, and actually, I think stool is in this as well, to really look at biomarkers. So that's measures of um, things in your blood or other systems that might be changing along with your pain that help us have that more objective output on what's happening. And as well as imaging, looking about are there changes in folks' MRIs or predictors on their MRIs that might help us understand the pain. And then lastly, drug pharmacokinetics, just learning more about how is the drug absorbed in your body and how does the body process this to help us figure out how it's acting. So a lot of assessments. Um, but I think time wise, I didn't put this in because of the time. I did want to say that it's 24 weeks is kind of this major part of the study, and it has that same safety analysis. So kind of over a while, we'll follow folks. I'm sorry, I don't have the actual dates, but if you want more specifics, you can always email me. I'm happy to chat. But if you are at all interested in joining the trial, learning more, like what would it actually involve, these are our two research nurses who work with our group, and you can call or email either one of them to get some more information. They can always share the consent form and information with you. It's not an obligation to join. So please feel free to reach out just to get, some more knowledge about this. And like I said, in terms of our lessons learned from the last trials, we've really taken to heart trying to minimize the travel burden. So if you are randomized to this arenumab arm, which is the injection we were just talking about, I believe it's only one visit in person. David, you can tell me if this is accurate. One visit in person where we can do a lot of the signing you up and getting you your first dose and some stuff, and then we can ship it to your house. So the medication would come in the mail. You don't have to come here to get it.
That's correct. Yeah, I went for my first appointment. I went in the infusion lab and waited there, and then the nurse gave me my first injection, and then I went home, and then they mailed me every month. I talked to Lauren, and
Yeah. So call on the phone to check in on
yeah,
you. And
check.
obviously, if something was up or you wanted to come in, we can accommodate you, but
Yeah,
only one required in person to really try to minimize this burden on folks.
I,
Unfortunately, with siltuximab, you do have to come in to get the drug more often because it's not an injection, it's an infusion. But we do have travel support thanks to some donors. We do have travel funding for folks. So if the barrier is primarily financial in terms of coming to receive siltuximab, we can hopefully offset that cost for folks and make it more affordable.
I'm hoping the Nubarab works for me, but if it doesn't, I know Dr. Plotkin said if I don't do the siltuximab, then I can't get back in it. So I might think about doing it, but it'd be hard every 3 weeks to come out there. I mean, the with the
Yeah.
travel help, that would be nice for me, but I work full time too, so that's
Yeah.
hard too.
We
And I pretty much
appreciate
ran out of
yeah,
right.
when people can make it, but we also understand that that doesn't solve the problem. It's not just about money, it's about time. It's about, a lot of people are in pain when they travel. And so as Dave said, if you get assigned to take siltuximab, these are things we're assigned, but like this is not a binding promise. If it doesn't work out for you, you're allowed to decline treatment. So for example, let's say you signed up for the trial, you were interested in this, and you got assigned to siltuximab, but then you say, I don't think I can do that. I can't actually come every 3 weeks. You're not bound to enter each of the studies just because you consented to the master trial. You also consent to these specific drug studies and say which of them you're willing to join. And same way we evaluate, is it each of these drugs safe for you to take? So it's kind of a two-stage consent where you consent first to the overall, I'm interested in this trial and I have pain. And then secondarily, you would be given information about each specific drug study and then decide whether that makes sense, both you and the investigator to join up.
Vanessa,
Okay.
it looks like there was a question from a while ago.
Oh, yeah.
It was
Sorry, I can't
about,
see the chat.
all right, it says, what about, it's going back, you're talking about genotypes.
Yes.
What about hybrid schwannoma neurofibromas tumors like mine? Are those being tracked like the genotypes?
Yeah, that's such an interesting question for anyone listening to this or who doesn't know. The most common tumor for schwannomatosis is schwannomas. It's how it got its name. It's a type of nerve sheath tumor, so the covering around the nerve. There's also, you might have heard of neurofibromas. That's the nerve sheath tumor that happens in neurofibromatosis type 1 or NF. But they're both nerve sheath tumors, right? Similar. So there's this thing called a hybrid nerve tumor, where under the microscope, a tumor looks a little bit like a neurofibroma, but also a little bit like a schwannoma, kind of an in between. And that's why we call it a hybrid. And I don't know of any studies that have actually looked at whether hybrid tumors are tend to be more or less painful than schwannoma tumors. I think it's the same, at least overall, some people have painful ones and some people it's not. The same way some people have painful schwannomas and sometimes you can have schwannomas that don't cause pain. Um, I don't think in these trials we're looking at it specifically because this doesn't involve surgery or biopsy. So this is treating pain kind of regardless of what tumors are causing it, or even if you think it's attributable to a specific tumor, because some people we know feel pain very localized to a tumor, and other people feel it more dispersed across their body. So these drugs are kind of treating that independent of the pathology of whether it's schwannomas or hybrid nerve tumors. But it's a really good question of something to look into and to see biologically, is there anything different? Or, when I showed that cytokine analysis in the lab, in the lab, is there anything different? I don't know of a study that's looked at that right now, but I'd be happy to look into it. And if I find something, and let folks know.
We have
Um,
another question here from Dale. Dale, you can speak up and ask your question if you want. Is our NF2 Schwann related patients allowed to join?
At
Looks
this
like not.
point in time, that's a good question. At this point in time, this is for non-NF2 related schwannomatosis. So this is only currently for people who have schwannomatosis that's LZTR1, SMARCB1, or you haven't had genetic testing and we don't know what it is. But it's not open currently for people with what used to be called neurofibromatosis type 2, what now is called NF2 related schwannomatosis. The primary reason being we don't have the same preclinical data. So all of this data that I'm talking about what drugs we think work for who, was all done in people with non-NF2 related schwannomatosis, kind of before the diseases got renamed and reclassified. But definitely an interesting point of study, knowing that they also have schwannomas and they also can be painful, although not typically at the same rate, or kind of it's not quite as high a percentage, at least what we think. It's not as high a percentage as other forms of schwannomatosis, that it could be there. So definitely a future avenue for study and something we are advocating for, to get this preclinical data that would help us, be able to add arms in the future. I
So
guess
I
the other thing we don't have as good of
yeah.
a job is everything I told you about before, the natural history study, these measurements. Unfortunately, I think it's just been a little bit ignored, or maybe ignored is too strong a word, but underappreciated in NF2 related schwannomatosis, because they tend to have, those folks have a lot of other problems, often with hearing and other things that dominate the conversation. But I do think there's a lot of folks who have said that they experience pain and might benefit from this. So I think it's an underappreciated area, but one we want to study and we want to get into the labs and understand, because it's a great point. It's the same kind of tumor. It's a very closely related disease. Why not add that into the mix?
So I'm curious, what about NF1 in pain and related schwannomatosis? I know they're different tumors, but obviously pain in NF1 tumors is very common, and
Yeah.
MEK inhibitors have really helped a lot with pain, but I assume that's a different mechanism, so the MEK inhibitors don't work the same.
Yeah, I don't know if anyone's actually studied a MEK inhibitor in schwannomatosis, knowing that the MEK inhibitors, like including selumetinib, have helped people with NF1. That is such a great point.
I
I
just assume
feel
maybe
like
it's a
someone must
different,
have tried this in the
they
lab.
must have thought of that. They're much smarter than me. I can't be the only one who thought of that. I would assume maybe it's a different mechanism because, it definitely helps with pain. I mean, my daughter's on it and it definitely helps with pain. And I was curious about that new drug, that Journavax that was approved recently. Um,
Which one is that?
it was just approved, back the end of January
Oh,
for
mirdametinib.
I
Were
don't
you
know.
saying?
Here it says Zeus Triga 9. I think the one that was approved as a non-opioid for pain.
Sorry. So we're not talking about NF. You mean
No,
like just a pain medication
it
that
was
was
approved for
recently approved?
people with, yeah.
I have not heard about that. I will look
I
into
mean, it
it.
was big news when I remember when they announced it a few weeks ago. I'm looking at it now. It targets pain signaling pathways in the peripheral nervous system.
Oh
So
yeah.
it's a non-opioid that was approved.
Would you mind putting that actually in the chat? I'm going to pass this on to all of our researchers. I mean, that's the awesome part about being at Mass General. And I just want to do this before we end today to thank everyone there for all the different studies I've talked about. We've had a wonderful, of course, want to thank Scott Plotkin, who leads our clinic and is the PI of all the studies, including the trials I've talked, but also Lei Shu, who is the co-PI of the Starfish trial, and she's brilliant work in the lab. So a lot of these suggestions about what we should be looking at, what drugs, what models, hybrid nerve sheath tumors, different genotypes. She and of course others in the NF community are kind of who we look to give us that data and tell us what they think will work that we can bring into humans. So I just want to thank everyone at MGH who's helped with these trials. Including Pam Walters, who has helped with how should we use these patient-reported surveys and measures, as well as these are some of the folks from Johns Hopkins, the University of Manchester, and NYU who helped with that natural history data collection. And then, of course, I want to shout out for the Starfish trial. We've had partners. So the one of the drugs has been provided by, this was from tanezumab, was the Pfizer provided that drug. Recordati, which is, I think, now EUSA Pharmaceuticals providing drug for the Starfish trial, and some funding from both the Children's Tumor Foundation and the congressionally directed medical research programs out of the DoD, which has provided funding for this trial as well. So a lot of support from different areas. And so I hope this is kind of an interesting story of kind of how we've evolved over the past, decade and a half and really gotten to this exciting place where we have clinical trials and it's not, we know what we're measuring. We have measures where actually have drugs we can test. And so I think all good hope from what we have here. Like I said, the Starfish trial is blinded. So until the 20 people enroll, we won't know for sure, whether this works or not, but I'm very hopeful. I think they're really exciting options. And so I think we are close, that hopefully within the next year we can recruit. So please, if you are potentially interested in participating in the trial or you know anyone, please feel free to share it. I'm really excited to see what that data, will show. And I think, we're really on the cusp of making a really important discoveries. We've had these 2 drug approvals for NF1 already. And so I think a lot of momentum in our community for both, NF2 and schwannomatosis to find something that, fingers crossed, we can say works more than that 30% of
Yeah.
currently available medication. So we want to help more people and for more durable, longer amounts of time.
Well, thank you, Vanessa. Are you able to see the chat now? Because
Yes, now that I'm not sharing, I can see. So I
okay,
will
because
look into this
Dale put something in there for you too.
Journavax and I will ask about looking into it.
And then
Dale
Dale
had
put a link.
another study of pain. Oh,
Yeah.
pain's cortical biomarkers. Yeah, lots of cool stuff about how the brain changes, how all sorts of things in your body, blood samples, stool samples, things that we can measure that around with pain, which will help us, one day hopefully predict better who is going to respond to certain medications. The ultimate hope for biomarkers is trying to be able to see that and figure out, oh, you have the kind of measurement or levels that mean maybe this drug is more effective for you. So I hope we get to that phase as well.
Exciting stuff. Exciting stuff.
Yeah, very
All right.
hopeful. So
Yep.
yeah, I hope that in a year or two, I mean, we should invite Scott back. It'll be his success with the trial, but hopefully, after we get to the 20 people in each study and everyone's treated, we'll unblind the results. So we'll figure out who was taking placebo and who was taking drug, and we'll be able to tell you the answer. Did it work? So really exciting that both of these will be, in the offing in the next year or two, hopefully, as soon as we can sign folks up.
Thank you. I think we'll formally end here, but I did want to say really quickly, if any of you, I have a sense a lot of you already know each other, but I know it's a small community. So if you don't know anybody and you want to exchange information, I'll leave this open for a minute or two to see if you want to share in a chat or even in the chat or actually talk if you want to share your information. So I'll give you about 2 minutes to do that. Or does everybody know each other?
Yeah, and I'm going to put my email in the chat too. Please feel free to reach out to me if you have any questions about what I presented, any ideas, any feedback at all. I'm really interested to hear what you guys have to say.
And lastly, if you want to put in the chat, if there's anything, I think it's, we have our info fair coming up and what's going to be about treatments, mainly focused on treatments, whereas last time was on research. But it's always hard to include schwannomatosis, and I feel really bad about that. But what we can do is do more of this type of thing. If we're going to do an info fair, then I'd like to, and we don't have schwannomatosis, I'd like to have at least a couple things that are online and work again with the NF Collective and everybody sharing it. So if you have some topic ideas, or questions that you want us to try and have a longer session to try and answer, you can put that in the chat right now, or you can email me. I'll put my email in there.
Yeah.
Maybe get, but you can always find me online. That would be helpful. So, shoot me information and we'll try to do, something else on schwannomatosis. All right, is that it?
Great stuff. Thank you.
Thank you guys for joining and spending time, and hopefully I'll have even better news to share next time we see each other.
Who's all going to the NF Summit?
I don't know yet. I'm usually there.
Weren't you at the last one, Diane? I think I met you there.
Yeah, I was in San Antonio and Scottsdale.
Yeah.
I will be here, and actually, so will Scott Plotkin. So please, if you see us, stop by. And if you have any in-person questions about the trials, we're happy to answer as well.
It's weird that I've never actually met you at Mass General all the years you've been working there, and I've been coming up there. I've seen everybody else but you.
She's probably
my office is in a different building, which is probably why. But next time, let me know. I'm happy to stop by. I love to, talk with people. As a researcher, I also want to thank you guys for sharing with me your experience. There are just so many things that are probably obvious to you, and that you're like, I can't believe it took them so long to figure out how much SWAT pain fluctuates. But the more I get to talk and hear what you guys have to say, the better our research is. So it's really a privilege to learn from what you guys are experiencing and sharing. And I took all of my notes, which I will be sharing with the team to follow up on.
And again, really let us know if there's anything else we should discuss. I know I see Dale a lot, so I can always ask him, but I don't know. Yeah, he's everywhere.
Dave and Bill are giving me a run for my money, so
I've seen them showing up a lot of places too.
No, you drag me kicking and screaming. Who you kidding?
All right. Well, everybody have a nice night, okay?
Yep.
Good night.
All right.
Thank you.
Bye.
Thanks,
Hopefully I'll see you all soon.
Bye.